Hi everyone, at this very today we are going to cover this topic from analytical chemistry. You can also find this topic in chemistry. So the topic name is chromatography, and these are some parts of chromatography. That is, we can cover these parts that are the concept of chromatography, partition coefficient, partition chromatography, TLC, paper chromatography, ion exchange chromatography, gel chromatography, adsorption chromatography, affinity chromatography, GLC, and HPLC. So today we are going to cover chromatography. In that we will cover partition and adsorption, and after that we will see that what will cover. Chromatography, so it is a perfect analytical technique for the identification, isolation and separation of compounds based on differences in affinity for a stationary phase and a mobile phase. So in chromatography, the stationary phase may be a solid liquid gel or solid liquid mixture, whereas the mobile phase may be a liquid or gas. What are the principle behind the chromatography? The principle of chromatography, the basic principle of chromatography is adsorption and partition. So when a mixture of compound is allowed to pass through stationary and mobile phases, the compound becomes separated according to their RF resolution front values. In here it is talking about that how the stationary phase and mobile phase get separated. So it gets separated with the because of RF resolution front. So RF is equal to distance traveled by the sample solute upon a distance traveled by the solvent front. Okay, you can repeat this one because this seems important. So the interaction during chromatographic separation substance include the following, that is partition and adsorption. So partition, what is partition? So when a solute is dissolved in two immiscible liquid of equal volume, the ratio of its concentration in two phases at a given temperature and equilibrium is called partition coefficient. Okay, we are revising it. That what I'm saying is that when a solute is dissolved in two immiscible liquid of equal volume, the ratio of its concentration in two phases at a given temperature and equilibrium is called partition coefficient. The partition chromatography and gas liquid chromatography mainly based on these types of interaction. So now we are covering adsorption. That what is adsorption? Here it is adsorption chromatography. So adsorption chromatography is the oldest type of chromatography technique. It makes use of mobile phase, which is either liquid or gaseous form. The mobile phase is adsorbed onto the surface of a stationary solid phase. Okay, adsorption chromatography is the oldest type of chromatography, okay, technique. It makes use of mobile phase, which is either liquid or gaseous form. The mobile phase is adsorbed onto the surface of a stationary solid phase. So adsorption chromatography principle. What is the principle of adsorption chromatography? So adsorption chromatography based on the analytical separation of a chemical mixture on the interaction of the adsorbate with the adsorbent. Adsorption chromatography based on the analytical separation of a chemical mixture based on the interaction of the adsorbent with the adsorbent. Adsorbate with the absorbent. Okay, so what is adsorbent and adsorbate? So a substance which is generally porous in nature with a high surface area of absorb substances on its surface by intermolecular forces is called adsorbent. Some commonly used adsorbent are silica gel N, silica gel H, alumina, etc. Okay, then we are going. This is adsorption chromatography. This is the diagram. Here solvent, it is XY mixture. It is silica alumina solvent. It is also solvent, and after that when we this solvent flows out of column. Okay, but in it is figure 1, it is figure 2. It is solvent, and when we mix silica alumina solvent is here, and X and Y is separated. Why Y is separated with X? Because Y moves down from column more quickly, so Y leaves the column. Okay, adsorption chromatography experiment, TLC. Take a clean means it is whole uh, experiment process of adsorption chromatography, and the example of adsorption chromatography takes, that is TLC. So take a clean and dry chromatographic jar to make sure that the environment of in the jar is saturated with solvent vapors applied to the walls. So add the mobile phase to the jar and close it. Maintain equilibrium. Mark the baseline on the adsorbent. Apply sample to TLC plate with the help of capillary tube and allow it to dry. So put the plates in the and close it. Wait until the solvent moves from the baseline. Take out the TLC plate and dry it. Okay, so this is the whole process of adsorption chromatography. Okay, so the application told here, that is not important, but if you want then you can take a screenshot from this. Okay, then we'll come on paper chromatography. So it was introduced by German scientist Friedrich Sakobin